Alright, so welcome to the tutorial session. So in today's session we are going to solve some problems related to optimizing drug target interactions. So far we have looked at a number of strategies that people use to optimize how the drug interacts with the target. And so this is obviously important because once the drug gets into the bloodstream and reaches the target, how does it interact with the target and how we can optimize those interactions in order to improve the efficacy of the drug. So the first question is this DU12290 which is the drug shown here was developed from this compound called as sultopride, and it shows improved activity and selectivity. So the question is, suggest possible reasons for this. Okay. So now let's look at these two structures. Sultopride is shown here, and in blue is the amide bond, and in the red is the carbon nitrogen bond. And so these are perhaps going to be some things that are important. And you also have a sulfate ester, and you have a methoxy group and a N-ethyl group. So these are the major functional groups that are present in the molecule. Now if you look at this DU12290, which is an analog, you see that a number of these functional groups are actually retained. So N-ethyl remains as N-ethyl, OME remains as OME, and this sulfite ester remains as sulfite ester or sulfone. So these functional groups are retained, whereas a new pyrrole ring is now been introduced. So once we look at the structure of the molecule, we can delineate these differences right away. So now one of the strategies that we have looked at previously is called increasing rigidity. Okay. So here what we do is we reduce the number of rotatable bonds. So the red bond that is shown here is the carbon nitrogen bond, is a freely rotatable bond. So in order to increase rigidity, we would want to convert that or reduce the rotation around that bond. So one way we can do it is to make it into a locked pyrrole ring. That is exactly what this analog they have done. So what this does is that it increases the chances of the active conformation being present when it enters the binding site. So we have already looked at this concept of active conformation. What the active conformation means is that of the infinite possible conformations, it is possible that a few of them or a subset of them are going to be the ones that are going to interact with the receptor binding site to induce the necessary conformational change. And so we want to get to a structure which is as close as possible to the active conformation, so that we can improve the drug target interaction. So in the case of the pyrrole, some of the conformations which are possible in sultopride are actually not allowed, and so therefore by doing this we are increasing the selectivity of the molecule to one particular receptor over the other. The next question is, explain the concept of a pharmacophoric triangle. So in order to address this question, let us go back and look at some of the lecture slides that we encountered previously. So let us go back to our hypothetical drug molecule, which is glipine. So it is very easy for us to draw out a two-dimensional pharmacophore. So here we have done this n number of times in the course. So you just identify the various functional groups and find out which are the ones that are going to interact with the receptor, or removal of which will result in a large decrease in the activity. So what this means is that there are certain groups in the molecule which are not that important, and there are certain groups in the molecule that are important. And we define the pharmacophore being the structure of the molecule which shows the activity. So here we have identified the pharmacophore in the following manner, which is it contains the two phenolic hydroxyl groups, the aromatic ring and the nitrogen atom. So these are the important parts of the pharmacophore. So in order to address the question of the triangle, now what we do is we can now, based on the structure of the molecule, we can align it in the form of a 3D pharmacophore. So here, instead of looking at the molecule as being in a plane, we start looking at the molecule in 3 dimensions. So we can measure the distance between, let us say, the aromatic ring and the nitrogen as shown here, and we can also start looking at the angle at which the molecule is out of plane. So here, for example, it is 18 degrees out of plane, and you can see here that the other two alcohol groups or phenol groups are also present. So now based on this we can specify the relative positions of the important groups in space, and as I mentioned, the angle is about 18 degrees from the plane of the ring. Now this helps us construct the pharmacophoric triangle. So what we can assume here is that these are the essential functional groups of the pharmacophore, and it is possible that they can share a what is known as a common pharmacophore. So, these 3D pharmacophores, of course, we will discuss it later when we are looking at molecular modeling, but the triangle approach is shown here, where what we start doing is we start constructing triangles. Okay. So, you can define these groups in as points in space. That means that you can call the van der Waals functional group as a point in space, the hydrogen bond acceptor donor as another point, and so on. And now one can start drawing triangles among these three groups, which are known as pharmacophoric triangles. Okay. And it allows us to compare across, let us say, library of molecules. So, when we define these triangles the way we have done it, it is now possible to take another library of molecules and look for these specific interactions being present in this format. Okay. And then we will hypothesize that these are likely to have some drug like molecules. So, here in our case, the phenol groups obviously can act as hydrogen bond donors or acceptors, and then the aromatic ring is going to participate in van der Waals interactions, and the amine can be a hydrogen bond acceptor, and of course, it can be an ionic center if it is protonated. The next question is, a cell surface receptor was identified as a target for new drug design. On the left, which is here, is shown the initial structure. So, what would be the characteristics of an agonist? Okay. So, here on the left is this molecule, and you can see here that there are there is a carboxylic acid, there is a hydroxyl group, and there is a negative charge, which is probably a substrate for an ionic interactions. So, what we would do is, if we have to design a molecule keeping this in mind, then we would place a group which has a positively charged species here, perhaps another positively charged species here, or a hydrogen bond donor over here, and something that interacts with the hydroxyl group over here. So, these are the three characteristics that we would look at and start screening for molecules. But the question is that later, while doing this study, they found that an additional hydrophobic region was discovered. So, now the question is, how will you revise your design? So, if you want to revise the design, let us draw out again the molecule that we are looking at. So, maybe we will have a positively charged species here, and then there is going to be another positively charged species or a hydrogen bond donor over here, and then there is going to be a molecule that is going to do hydrogen bond interaction with the hydroxyl group. So, let us say we come up with this type of a molecule. Once we discover this extra hydrophobic group, then the logic that we would follow is to introduce alkyl groups. So, by introducing alkyl groups, what we can do is to sample for this hydrophobic interaction. Since the alkyl groups are involved in hydrophobic interactions, so we would be able to improve the binding with this receptor. So, this is called an extension strategy, and it works by trying to improve the activity or improve the number of bindings that we can achieve. The next question is, selective beta blockers were developed by replacement of the aromatic ring in adrenaline with a naphthalene ring system, as shown below. This resulted in a compound that was able to distinguish between two very similar receptors, that is the alpha and beta receptors of adrenaline. Question is, explain. So, in order to address this question, what we need to do is we need to look at both of these structures carefully, and one possible difference, I mean one major difference between these two molecules is that the two hydroxyl groups are gone from adrenaline, and you have now a situation where you can have van der Waals interaction. So, this is something that is distinct between or different between the two molecules. So, therefore, it is likely that there is some van der Waals interaction that is going to be in play, which is not present in the molecules on the left. Another possible explanation is that if the receptor is going to interact with this ligand, then there may be something that is preventing sterically the interaction as shown here, right. So, what may happen is that one of the receptors is going to bind in a particular way and it fits perfectly, but the other receptor may be binding like this, and so what happens is that you have a steric clash, which is going to result in some selectivity for one receptor over the other. The next question is, explain the concept of extension of a structure. We have already looked at this in the tutorials, but let us revisit this. Extension is nothing but addition of another functional group or a substituent to the lead compound in order to probe for extra binding interactions with the target. So, here in the example that we looked at, we had three interactions that were already present, but now by adding additional functional groups, we may be probing for stronger binding. Okay. So, the basic question that we ask in this extension of structure strategies, are all the binding sites being accessed? So, if the answer to that is no, then we can keep adding structures to it to sample for or improve the binding. So, here is the pictorial representation. So, let us say a receptor site has these 4 functional groups which are going to interact with your target. Then what we could do is we can design a drug, let us say, which interacts with 3 of these groups in a particular way. Now, since we are aware or we have some idea that there could be an additional binding site, we can use what is known as an extension tactic. So, we can add an alkyl or an aryl alkyl group, so that the next group that is shown here can also be binding to the target.